Every cell in the human body contains the same DNA. Every human has different varieties, different variants of each of their genes. This is due to mitosis and meiosis. Mitosis is going to give every cell the same DNA. Meiosis is going to ensure variability in DNA between individuals. Let's take a look at these processes. In this lecture, I'd like you to define DNA, chromosome, genotype, phenotype, gene, allele, mitosis, meiosis, cytokinesis, haploid, diploid, homozygous, and heterozygous. That's a lot. List and define the phases of mitosis and meiosis. And by define, I mean what's going on during these. So I'm going to keep this very short, very sweet, and just try to go through the phases one by one as simply as I can. There are other internet resources that go into more depth, less depth, or rapid out. Feel free to avail yourselves of those resources as well. The cell cycle. So cell division is what we're into today. And cell division is the process of making new cells starting from one cell. So you have a cell, it's going to grow, it's going to do metabolism, it's going to make new DNA, and it's going to prepare itself, and it's going to divide. And then you have two cells. Okay, and that's what we call the cell cycle. From mitosis to mitosis. So let's look at that M down there. The M is mitosis. After mitosis, you have two new cells, and each of those cells will go through normal metabolic processes during what's called the G1 phase. There's a checkpoint in there that is going to say, well, do we need to divide and make two cells? And if the answer is yes, then that cell will enter what's called the S phase, where it synthesizes new DNA. So it makes a copy of each DNA molecule during S phase. During G2 phase, the cell is going to grow a little bit more, and it's going to prepare itself for M phase, which is going to be another mitotic division. Mitosis is about dividing up the same DNA and moving chromosomes from one cell to another. Well, one cell to two cells. So let's look at DNA. DNA is these long molecules of nucleic acids in a double helix packed into what is called chromosomes. The structure of DNA molecule is this double helix of paired nucleic acids, and it's a very long molecule. This molecule is kind of packed up in what's called a chromosome, which is when you wind DNA around histones, wind the histones around each other, make this huge thing, well, huge compared to the original strand of DNA, but it's this chromosome. So a single chromosome can just be one of those, two of those four arms you see there on your replicated chromosome, but it's really just one long DNA molecule. And when you replicate that DNA molecule, you get a replicated chromosome made of two sister chromatids. They're sisters because they look, well, they're exactly the same. This isn't Anna and Elsa. These are twin sisters of this replicated chromosomes. You have sister chromatids, two chromatids, one replicated chromosome. Let's look at these phases of mitosis here first. So what we're going to do in mitosis is we're going to take those replicated chromosomes and we're going to put them in two new cells. Interphase is not a phase of mitosis so much as the time between mitotic events during which the cell is going to do metabolism, synthesize new DNA, and prepare itself for division. The first thing it does in preparing itself for division, besides synthesizing new DNA, is to pack that DNA up in prophase. Prophase is about packing up the DNA. If we're going to separate that DNA, we have to get it out of the nucleus, and this is done by dissolving the nuclear membrane. So prophase, pack up the chromosomes, break down the nuclear membrane. Prometaphase is going to connect the DNA, the chromosomes, to this mitotic spindle, which is going to help move them around so we can split them apart. Let's look at that mitotic spindle. Don't memorize these terms here, but do know that you have these centrosomes, these things on the other side. The other sides of the cells are going to pull towards themselves, and this aster is connecting to all of these chromosomes, and it's going to move them to the middle of the cell, the metaphase plate during metaphase. It connects to chromosomes. That's what's called a kinetochore, which you see down here is the center of the chromosome, right where the two sister chromatids are attached to one another. That's the kinetochore, and that's where the mitotic spindle is going to kind of join them, or going to connect to them. Oh, and you see that there's two mitotic spindles, one attached to each side of each chromosome. So one attached to each sister chromatid for each chromosome. So during metaphase, these chromosomes are lined up in the middle. M for metaphase, M for middle. They're lined up here, and that's going to help us to separate them to different sizes, which occurs during anaphase. During anaphase, these chromatids will separate. Now, when they're separated, they become individual chromosomes because they are still that replicated DNA. So as they separate, they're going to move to the opposite sides of the cell. And when they get there, we begin telophase. Telophase is essentially prophase in reverse. The nuclear membranes begin to form around each new nucleus, and the chromosomes begin to kind of decondense and allow access to the DNA again. You're going to get two new nuclei, and then you're going to have two cells. But mitosis is the division of DNA. We also need to divide other things, and we're going to be dividing here what's called the cytoplasm and the organelles during cytokinesis. Cytokinesis divides up the cytosol, divides up the organelles, so that both new daughter cells get mitochondria, get a Golgi apparatus, get an ER. All of these things are going to be found in both new daughter cells because cytokinesis is going to divide up these things after mitosis is done. And what you have here is you have two new daughter cells, both with the same DNA as their parent. You have now, through mitosis, created two new cells that have the same DNA as their parent. End mitosis, begin meiosis. In meiosis, the goal is variability. The goal is not to make two new daughter cells that are the same, but to make four new haploid cells. So, in order to understand this, we're going to have to look at alleles. Alleles are variants of genes. So we're going to talk about variation. We're going to talk about alleles. Let's say we have a plant. So this is a pea plant, and it's going to have two alleles that are the same. This is the tall alleles, or it has two short alleles. These are both homozygous plants. Two plants. Let's say that these two plants were crossed. So one's going to pollinate the other, and one's going to bring a big T, and one's going to bring a little t. This offspring is heterozygous. So a heterozygous organism has two different alleles for a character, height, and its heterozygous here has the two alleles, and it's going to express one but not the other, but we're going to get to that in the next lecture. So homozygous has the same, heterozygous has different, and what we're going to see is, let's say you've got the big T from its father, small t from its mother. It kind of looks like this. It has homologous chromosomes because the same gene, the height gene, is on chromosome 1 for this plant in my hypothetical example. The height gene is for chromosome 1, and you can have the T, big T allele, tall allele, or you can have the little t allele, the dwarf allele, to make short plants. These are two alleles for one gene, giving it a genotype, and the genotype is, well, what kind of big T, little t, little t, little t, that's your genotype. Your phenotype is that this plant is actually going to be tall. So genotype is your genes, phenotype is what you see, and we're going to get into that more during the next lecture as well. What I want you to look at here is the homologous chromosomes. All diploid organisms get one chromosome from their dad and one chromosome from their mom. How do you know if they're diploid? Do they have a dad and a mom? Simple as that. Are they coming from a cross between two individuals? If they are, they're probably diploid. So let's look at meiosis in two phases. What we're going to do is make haploid alleles from a diploid cell. We start with this diploid cell. It has a pair of homologous chromosomes, one from dad, one from mom. Let's say it has those T alleles, right? You're going to replicate that in the S phase, which is going to make two sister chromatids for each one of those chromosomes. In meiosis 1, they're going to separate the homologous chromosomes. So for all the DNA that came from dad goes one way, all the DNA that came from mom for this chromosome goes another way. That is separating the homologs in meiosis 1, leaving haploid cells that still have those duplicated chromosomes. Meiosis 2 is going to make the sister chromatids separate. So that's going to be very similar to what you saw in mitosis. We're going to play that similarity pretty well, making for 4 haploid cells with those unduplicated chromosomes. That's the goal of meiosis. So let's take another look at this, and then I want to take a closer look again. So meiosis is going to be divided up into meiosis 1 and meiosis 2. In meiosis 1, get all the way up here. In meiosis 1, we have prophase 1, metaphase 1, anaphase 1, and telophase 1. During these 4 phases, there's prometaphase 2, we don't focus on that. But during these 4 phases, we're going to see the homologs separating. So in prophase 1, the homologs are actually going to pair up with one another. They're going to exchange some DNA. We're going to come back to that. In metaphase 1, the chromosomes line up on the metaphase plate, but this time, instead of lining up individually, each one pairs up with its homolog. During anaphase 1, the homologous chromosomes will separate, leaving 2 haploid cells that occur during telophase 1 and as cytokinesis occurs and pinches them off. That's meiosis 1. Meiosis 2 is going to separate the sister chromatids. During prophase 2, break down the DNA, break down the nuclear envelope, same as prophase of mitosis. Metaphase 2, line them up in the middle of the metaphase plate. Anaphase 2, separate those chromatids. They're going to become individual chromosomes. You're separating the sister chromatids there. And then telophase 2, you will have 4 haploid gametes. So in meiosis 1, we're going to see 3 processes. And I want you to look at these 3 processes as we move through. The first one is during prophase 1, and that's called crossing over. The second one is going to be during metaphase 1. And that's going to be called independent assortment. And next, remember that in anaphase 1, the homologs separate. So the homologs will separate there. Okay, let's look at this closer. In prophase 1, you see these 2 homologs are going to form what's called chiasmata. A chiasma is a crossing over. Essentially what's going on, Cat, is these two of the sister chromatids are going to exchange DNA with one another. So that big T, little t allele, they're going to be swapped over for 2 of these arms so that now the mother is going to have part from the father and the father is going to have part of it from the mother. That's going to make for a new combination of alleles. So if the mother had little t, but also had an allele for purple flowers, and the father had big T, but also had an allele for white flowers, now you're going to get a combination that is new, which is big plant, white flowers, or purple flowers. You're going to get them basically swapped over. And that swap over creates new types of variation. So new types of variation in prophase 1 due to crossing over. In metaphase 1, how you pair up the homologs is independent of how other homologs are paired up. So you're going to move them around and perhaps put the father on the right, mother on the left, or perhaps put the father on the left, mother on the right. You randomize it for each of the chromosome types. So you have 23 chromosome types in the human body. So there are 2 to the 23rd different combinations that you can make of gametes just based on how you assort them, because the assortment is independent. That's independent assortment, and it means each haploid cell at the end of this is going to get a novel combination of maternal and paternal chromosomes. In anaphase, you keep those sister chromatids attached and you separate the homologs, which will give rise to 2 haploid cells in telophase 1. So that's meiosis 1. We're making cells haploid, assorting the chromosomes independently, and there's some crossing over. Let's look at meiosis 2. In meiosis 2, what we're going to have is the sister chromatids separating. It's that simple. It's very much like mitosis. In prophase 2, you have the lining up, the breakdown of the nuclear membrane and the packaging, if they're not already packaged, of the chromosomes. Same as prophase of mitosis. In metaphase 2, you're lining them up on that metaphase plate. Very similar. In anaphase 2, you break apart the sister chromatids. The difference here is that we're doing this in a haploid cell as opposed to a diploid cell. And we're doing this with recombinant DNA that has been, affected by crossing over, not with non-recombinant DNA that has not been affected with crossing over. So this is the difference, is that the difference between meiosis 2 and mitosis is that meiosis 2 comes after meiosis 1, effectively. In telophase 2, you have 4 haploid cells. That's the end result. 4 haploid cells, each of which is going to have a new combination of chromosomes from mother and father, and each of which is going to have new combinations of alleles due to crossing over. So that's mitosis and meiosis in a very bulleted kind of way. Feel free to avail yourselves of other resources online. I said there's a rap for it, there's a line dance for it. There are a whole bunch of different videos, all of which tell the same story of mitosis and meiosis. Mitosis makes 2 new daughter cells the same as the first cell. Meiosis makes 4 haploid daughter cells that are different. There we go.